When conducting passaging cells technique, make sure to follow strict aseptic technique. This includes disinfecting the interior surfaces of the flow hood with 70% ethanol, as well as anything else you place into it.
Visually assess confluency of the flask. If it's over 80%, the cells will need to be split. Remove the media from the 25 cm squared flask containing the cells using a pipette, being careful not to disturb the cell layer. Using a pipette, add 2 milliliters of trypsin to the flask, allowing contact to the cells for 10 to 30 seconds. Be careful not to pipette the trypsin directly onto the cells, as it could damage them.
Once the 2 milliliters of trypsin have been added to the flask, verify using spectroscopy that none of the cells have detached from the flask wall.
Remove 1 milliliter of trypsin and rock the remaining amount over the bottom of the flask. This will allow for grains of sand to be seen when held towards the light. Verify using spectroscopy that all cells have become detached from the wall of the flask. Next, deactivate the trypsin using 2.5 milliliters of culture media with FCS. Pipette roughly 1/2 milliliter of cells into a microcentrifuge tube for a cell count. Combine both a 10 microliter cell aliquot from the previous tube and a 10 microliter aliquot of trypan blue into another microcentrifuge tube and dilute appropriately. Pipette 10 microliters of the cell suspension into the opening of either of the two chambers of the counting slide and measure using the electronic cell counter. Once the cell count has been obtained, use the appropriate amount of flasks to separate the cells from the original flask. Add the appropriate amount of culture media, increasing the original flask volume to 7 milliliters, and separate equally into the additional flasks. Finally, label each of your flasks properly with cell type, passage number, date, and an indicating symbol. Once this has been done, place each flask into the incubator for a 24-hour period.